Hey everyone, so today we'll be looking at reconfiguring coronaviruses to fight cancer. Let's delve into a bit of history. Back in 1904, Professor George Dock observed something intriguing. A 40-year-old woman with acute leukemia saw a brief improvement in her condition after catching the flu. Now it's worth noting that George was also the same person who prescribed arsenic solutions to his patients. But remember, this was in the early 1900s. In 1953, a fascinating case emerged. Physicians reported that a 4-year-old boy with lymphatic leukemia experienced a remarkable improvement when he contracted chickenpox. Before the infection, he had an enlarged liver and spleen. However, within days of developing the variola rash, his spleen and liver returned to their normal size. Sadly, this remission only lasted a month, and his leukemia rapidly progressed, ultimately leading to his death. And so, the idea of harnessing viruses to fight cancer has been around since the 1850s. It's been observed that in some cases, when immunocompromised patients contract natural virus infections, their tumors can regress. The general concept is that cancer cells persist because they evade the immune system. But that also means when they don't communicate well with the immune system when they are attacked by viruses. Initially, researchers explored various natural viruses for their potential in cancer treatment. In 1949, they experimented with the hepatitis B virus, followed by an early passage of the West Nile virus in 1952, adenovirus in 1956, and the mumps virus in 1974. However, many of these attempts were discontinued due to concerns about their effectiveness and safety. Subsequently, scientists began investigating a wider range of viruses for their anti-cancer properties. One of the favored viruses for cancer treatment is the herpes virus. Its versatility lies in its ability to infect various types of cancer cells. Plus, we have acyclovir as a backup to take care of any issues that may arise from herpes infection. Despite about half the population having antibodies against herpes, the virus can still evade host immunity, making it particularly favorable for therapy. Viruses in general trigger the host immunity, so oncolytic viruses aren't very useful if it gets neutralized before it kills cancer. So I thought, why not use coronaviruses? Okay, so let's steer clear of viruses like SARS, MERS, and SARS-CoV-2, and focus instead on HCoVs, which stands for human coronaviruses. These are viruses responsible for common colds. It's a safer choice. But why coronaviruses? Well, the protection they offer against infection doesn't last very long, and it doesn't shield us against other strains. Plus, there's a risk of reinfection from the same strain within just 6 months. But since the discomfort they cause is minimal, and that our body does not sustain long-term protection, these coronaviruses can be repurposed relatively quickly for oncolytic virus therapy. In selecting our viruses, I've compiled a table outlining some key differences. It's important to note that while there are some variations among the different coronaviruses, they also share many similarities, including features seen in COVID. Back to our selection criteria. We'll stay away from HKU1, which can induce febrile seizures, and NL63, which is known for causing hypoxia and croup. The neurotropism makes it potentially useful for brain cancers without too much downsides. Next, let's consider the entry points. 9-O-axia infects the respiratory tract and beyond, while APN targets epithelial cells in both the respiratory and gastrointestinal tracts. for those curious, COVID also utilizes the ACE2 receptor, which the NL63 also uses. Ultimately, I decided on the HCOV 229E. Its ability to infect both the respiratory and gastrointestinal tracts makes it well suited for targeting tumors in various locations. In the early stages of research, efforts focused on ensuring that viruses could effectively target cancer cells. One approach involves targeting CD155, a protein overexpressed on the proteins of many tumor cells. To achieve this, we will use VP1, which is derived from polio. This is also why some scientists have explored using polio to target cancer. Additionally, we'll insert the MGCSF gene, which stands for Human Granulocyte Macrophage Colony Stimulating Factor. This gene helps attract immune cells to the tumor site and also reduces the tumor's access to blood and nutrients. And the gene will roughly look like this. I've taken the liberty to name it RHCOV 229E VP1 MGCSF. We're essentially done and just need to insert the actual genes into the virus. But otherwise, we should talk about the dangers of this. As we're aware, the common cold coronaviruses spread through water droplets. By modifying the common cold virus for cancer treatment, we've essentially made it more hazardous. If something were to go wrong, the consequences could be significant. However, on the flip side, if our approach proves successful, pharmaceutical companies may not find it profitable if people will be cured of cancer for free after contracting our modified virus. Otherwise, that's all for today. If you're interested in understanding how the virus design process, check out my video where I recreated the Ebola vaccine. If you want to know how the COVID virus works, let me know in the comments. Thank you for watching.